All right, so today we're going to be taking a deep dive into the world of microRNAs.
These tiny little pieces of RNA that are like, I think people are going to be surprised to learn how much they actually impact like our day-to-day lives.
Yeah, well, when I first heard about microRNAs, I have to admit, I kind of thought, like, how can something so small be so important?
Yeah.
But it turns out they're involved in pretty much like every process in our bodies.
They're like little puppet masters, you know? They
Yeah.
control what our genes actually do.
Exactly. And what's even crazier is that, like, the first microRNA was discovered in a worm.
Yeah.
A worm.
A worm. It's a round worm called C. elegans.
Oh, wow.
And scientists were studying how genes are regulated in these worms and stumbled upon this whole other mechanism of microRNA-mediated gene silencing. And it was such a big deal that it won the Nobel Prize in Physiology or Medicine back in 2024.
Wow. A Nobel Prize from studying worms. That's incredible.
Yeah.
So how do these microRNAs actually work? I mean, we're talking about something that's tiny. Like, how do they control something as complex as a gene?
So think of it this way. Your DNA has the blueprints for all the proteins your cells need to function.
Okay.
But those blueprints need to be read and translated, and that's where messenger RNA comes in, or mRNA.
Okay.
And it carries the genetic code from the DNA to the ribosomes, which are like the protein factories of the cell.
So the mRNA is like the delivery guy taking the blueprints to the factory floor.
Exactly. And that's where these microRNAs come in. They're like little sensors.
Okay.
And they each have their own unique sequence that allows them to bind to a specific messenger RNA.
So almost like a lock and key type of
Yeah,
thing.
you got it.
Yeah.
And once it binds to its target mRNA, it can either block it from being translated into a protein or even target it for destruction.
Wow. So it's like intercepting the blueprints before they can even reach the factory floor.
Precisely.
That's wild. So we've talked about like but I'm curious about the how. Like, how are these microRNAs made?
So it's actually a very fascinating process. And we actually have a research paper by Scott M. Hammond that breaks down how these things are actually made.
Oh, cool.
And it's like a bustling cellular factory.
Really?
Oh, yeah.
That's kind of a fun way to think about it.
Yeah, it's very intricate.
Okay, so walk me through the cellular factory.
Okay.
Where do we start?
So we start in the nucleus of the cell,
Okay.
where our DNA is stored. And a microRNA will start its life as a much longer RNA molecule called a primRNA.
A primRNA.
Yeah.
Okay. And that's like the raw material.
So it's like this long sheet of blueprints that needs to be cut down to size.
Exactly. And just like in a factory, we have specific tools to do this. And the first tool is an enzyme called DROSHA.
DROSHA.
Yeah.
Okay.
And it works with a protein complex called DGCR8 to make the first cut, trimming that primRNA down to a smaller hairpin-shaped molecule called a preMRNA.
DROSHA, DGCR8. These are some pretty interesting names for molecular tools. Yeah, scientists like to have a little fun with the names. It's kind of like it's gone through its first round of processing in the factory, but it's still got to be transported out of the nucleus into the cytoplasm, where those protein factories are, the ribosomes.
So you need a delivery truck to get to the factory floor.
Exactly, and that's where exportin 5 comes in.
Exportin 5.
Yeah, it's the special protein that acts as a transporter, and it recognizes that preMRNA and guides it out of the nucleus through a channel called the nuclear pore complex.
Oh, wow. It's like a whole little highway system inside the cell.
Oh, yeah, very much so.
So the preMRNA is out in the cytoplasm now,
Right.
ready for the next stage of processing.
Yeah, exactly.
What happens next?
Well, this is where our friend Dicer comes in that we talked about earlier.
Okay.
He's just waiting in the cytoplasm, ready to make the final cut.
Right.
And Dicer, it trims off the loop of that hairpin structure, leaving us with a short, double-stranded RNA molecule about 22 nucleotides long.
Okay, quick question. Can you remind me what exactly nucleotides are again?
Oh, yeah, so nucleotides, those are like the building blocks of DNA and RNA.
Right.
Like the letters of the genetic code.
Okay, got it. So we've got this tiny double-stranded RNA molecule.
Yeah.
It's been through like a whole journey at this point.
It has.
To get to this point.
It's been through a lot.
But how does it actually silence a gene?
So one strand of this double-stranded RNA molecule is actually the mature microRNA, and it joins forces with a bunch of proteins to form this complex called RSC.
RISC.
Yeah, it stands for RNA-induced silencing complex.
Ooh, that sounds important.
This is like the workhorse of the gene silencing process.
Okay.
It uses the microRNA as a guide to find and bind to specific mRNA molecules that have a matching sequence.
Oh, so it's like It's searching for its perfect match. It's
Exactly.
like finding an address in a huge city.
Precisely.
Wow.
And once RISC locks onto its target mRNA, it can either physically block the ribosome from translating that mRNA into a protein,
Oh, wow.
or it can even initiate the destruction of the mRNA altogether.
Wow, so these microRNAs really are like the puppet masters here.
I know,
So
right?
they control which proteins get made and which ones don't.
Yeah, and it's really amazing how precise this whole system is.
It really is, but it's not about completely turning genes on or off, right?
Right, it's more about fine-tuning that protein production,
Okay.
like adjusting the volume knob on a complex orchestra.
That's a great way to put it.
Yeah.
So how does this fine-tuning play out, like in our bodies? What are some examples of how microRNAs impact our health?
Well, the Hammond paper gives some really interesting examples.
Okay.
For example, there's MIR-208.
Mir 208.
Yeah, and it plays a key role in heart health.
Okay, so we're going from tiny molecules to something as vital as our hearts.
Our sex.
I like it. This is where it gets really interesting for me.
So MIR-208, it regulates a protein called MYH7.
MYH7.
Yeah, which is crucial for the heart muscle's ability to contract and adapt to stress.
Okay.
Without enough MIR-208, the heart can become less efficient, potentially leading to problems down the line.
So if Mir 208's like the conductor, making sure everything's working right, what happens when these microRNAs aren't working the way they should?
Well, that's when things can go wrong in a variety of ways. For example, let's look at MIR-122.
Okay.
This one's really interesting. It actually plays a role in how the hepatitis C virus replicates.
Wait, so you're saying the virus uses our own microRNA against us?
Yeah, it sounds crazy, right?
That's kind of scary.
But it's a really fascinating example of just how resourceful these viruses can be.
Wow.
The hepatitis C virus actually needs MIR-122 to stabilize its RNA,
Oh, wow.
essentially helping it hijack our own cellular machinery to make more copies of itself.
That's incredible. So we're learning all this stuff about microRNAs, how they work, all that, but like, can we use that knowledge to actually develop new treatments?
That's the million-dollar question, right?
Yeah.
And it's something that scientists are very, very excited about. And we're talking about the potential here for microRNA-based medicine.
MicroRNA-based medicine, what would that even look like?
So imagine this. You go to the doctor for a checkup.
Sure.
And a simple blood test could reveal whether or not certain microRNAs are out of whack,
Oh, wow.
potentially indicating a problem before you even have symptoms.
So early diagnosis just by analyzing your microRNAs.
Exactly.
That would be incredible.
And beyond diagnosis, researchers are even developing ways to manipulate microRNA levels as a form of therapy.
Really?
Yeah, so for example, if someone has a condition where a specific microRNA is deficient, like we were talking about with MIR-208 in the heart,
Right.
we might be able to develop a microRNA mimic, which is essentially like a synthetic version of that microRNA to restore its function.
So giving the body back the tools it needs to heal itself.
Exactly. And then on the flip side, for microRNAs that are overactive in certain diseases, like some cancers, scientists are working on microRNA inhibitors,
Yeah.
which would act like a mute button, dialing down the activity of that specific microRNA.
It's amazing to think that we could be on the verge of these like really targeted treatments.
I know, it's super exciting.
But I imagine there's still some challenges that need to be figured out.
Oh, absolutely. One of the biggest hurdles is figuring out how to deliver these microRNA mimics or inhibitors to the right cells in the body.
Right. You don't want them to go everywhere.
Exactly, and without causing unwanted side effects.
Right,
So
yeah.
it's like delivering a tiny package to a very specific address in this bustling city of cells.
So how are scientists trying to tackle that delivery problem?
They're getting really creative.
Okay.
One promising approach uses tiny nanoparticles.
Nanoparticles.
Yeah. They act like protective capsules, shielding the microRNA from degradation and helping to deliver it to the target cells.
Wow. So we're talking about like designing a microscopic delivery system.
Exactly.
Wild. This has been incredible. I feel like we've only scratched the surface of what there is to know about these microRNAs.
It's true. And to think it all started with a tiny little worm.
I know, right? Who knows what other incredible discoveries are waiting to be made in this microscopic world inside ourselves? Well, that's something to think about, for sure. For all our listeners out there, thanks for joining us on this deep dive. Be sure to stay tuned for more fascinating insights into the world of science and technology.